Hello, my name's Roger Watson and welcome to this session on protection. We live in a hostile environment and we require protection against oxygen, injury, poisoning, extremes of heat, dehydration and infection. Let's look at these in turn. It may seem strange to say that we require protection against oxygen because oxygen is essential for life. The air we breathe is in fact only 20% oxygen. We're adapted to live in that percentage. Higher percentages of oxygen are damaging to human tissues. The majority of the air we breathe is nitrogen, and that's inert. In other words, it doesn't react with substances in the body. In fact, oxygen is highly toxic, and if it's breathed at higher concentrations, it leads to severe effects as follows. It has an effect on the eyes, affects vision. In the long term, it leads to cataract formation, can lead to seizures in the central nervous system, leads to muscle twitching, jerky breathing, coughing, pain, and respiratory distress syndrome. It's very rare, of course, that we do have to breathe oxygen in high quantities. Sometimes therapeutically it's required for a short time, and airline pilots, test pilots, when they're testing planes at very high altitudes, also use high tensions of oxygen which are pumped into the lungs in order to accommodate for the low levels of oxygen and the difficulty they have in breathing, but this can only be sustained for a very short time. Oxygen, when it reacts with some substances in the body, produces active oxygen species, specifically superoxide and hydrogen peroxide. These are very damaging to DNA and to cell membranes. And we have mechanisms in the body to protect against these toxic effects of oxygen at normal levels of oxygen. So even in normal levels of oxygen we're suffering from toxic effects, but thankfully we have mechanisms to protect against those toxic effects. And this diagram summarises the mechanisms for protecting against active oxygen species. On the left you see, as a result of aerobic metabolism, a normal process, the production of O2-, which is superoxide. This is then broken down by enzymes. They're called SOD, or superoxide dismutase enzymes. There are various forms of this associated with different metals in the body. The superoxide is broken down along with hydrogen into hydrogen peroxide, or H2O2, and this can be broken down by two enzymes, catalase and glutathione peroxidase, into water and oxygen, which of course are not harmful, and that ensures cell survival. The danger with the hydrogen peroxide is that in the presence of iron it can be converted into hydroxide ions, or OH dot ions. These are also highly active oxygen species, and these are very damaging to cells. So the superoxide, the hydrogen peroxide and the hydroxide ions are all damaging to cells, and we have mechanisms for removing these either directly, or in the case of the hydroxide ions, indirectly, by removing the substrate, the peroxidase from which they're formed. With aging, we become less able to protect ourselves against the harmful effects of oxygen, and this may well be why we see some of the signs of aging, like wrinkling of the skin, for example, and other effects such as the increase in cancers and the remainder of the aging process itself. At least part of that may well be due to the toxicity of oxygen and our decreasing ability to protect ourselves against it. Of course, this has spawned a great industry in foods and facial creams which are claimed to be antioxidants. The antioxidant industry, but I have to say there's little or no evidence that any of this works. Essentially, the aging process cannot be reversed. Next, let's look at reflexes, which are also protective mechanisms. A reflex is an automatic, instinctive, unlearned reaction to a stimulus. For example, if we step on a drawing pin, we automatically pull our leg away. We may well feel pain as a result of doing that, but in fact, we pull our leg away before we feel the pain. A signal is sent to the central nervous system which causes us to pull our leg away from the source of the pain. That's a reflex. To protect against chemical injury and sometimes infection, we have a protective mechanism called vomiting. It's the reflex act of ejecting the contents of the stomach through the mouth, something that we will all have done at some stage in our life. As this slide shows, there are various mechanisms which can cause us to vomit. Vomiting is mediated via the vomiting centre in the medulla. So at the higher cortical centres, even the anticipation or the memory of something that makes us sick or makes us afraid, or the sight of something that makes us sick, or the smell, or even suffering pain, these can have an effect through the higher cortical centres on the vomiting centre. But also from the stomach or the intestines, things like chemotherapy, surgery or radiotherapy, or indeed poisoning or infections like gastroenteritis, these can have an effect directly on the vomiting centre or indirectly by chemoreceptors elsewhere in the body, and also substances like opioid drugs can make us vomit. And then the labyrinths of the inner ear, which detect and help us to maintain balance, these can be affected in infection or in surgery, and these can also have an effect on the vomiting centre. So vomiting is a reaction to various stimuli. And essentially in vomiting, the diaphragm is pushed down, as shown on the right hand side, and the ribs contract to put pressure on the stomach, and the contents of the stomach are ejected through the esophagus and through the mouth. The next aspect of protection I want to look at is the skin. The skin protects against physical damage, dehydration, extremes of heat and infection. And the skin, in fact, is the largest organ of the body. Here you see a body that's been flayed, the skin's been removed, and the skin is easily seen there as being the largest organ in the body. The simple structure of the skin is that the outer layer is the epidermis, which is covered by a dead keratinized layer of cells. Underneath that the dermis, and below that the hypodermis. The hypodermis is a fatty layer where the blood vessels travel along under the skin. The dermis contains the various organs of the skin, which we'll look at in a minute, and the epidermis is the outer layer. One of the obvious things about human skin is that you can see hair projecting through most of the skin. This cross-section of the skin shows various things from the left to the right. From the left we see a sweat gland. Sweat glands secrete sweat, which is really water and uric acid, onto the surface of the skin. This gets evaporated, helps us to cool down. You can see the pores there, and these are the holes where the hair projects down into the hair follicle. In the follicle you can see there's a sebaceous gland. This secretes an oily surface up through the hair onto the skin. This affords us some protection, a natural moisturizer. And there's also a piloerectile muscle, This doesn't serve any function in humans, it's a vestigial aspect of our evolution, but when we're cold or when we're frightened, this causes the hairs to erect, giving us that appearance of goose bumps. And there are also nerve endings in the skin. A major function of the skin is thermoregulation, and you may want to look at this Bozeman Biology YouTube link on thermoregulation. Thermoregulation is under the control of the temperature regulating centres in the hypothalamus. It's a rather complicated diagram, but what it shows is at the right hand side, let's start at the right hand box, the stimulus, which is a decreased body temperature. This is sensed in the brain, in the hypothalamus, which is a set point for body temperature, and if the body temperature drops, this activates heat promotion, and the two aspects of heat promotion that are promoted here are shivering of the large muscles, which causes us to generate heat, and constriction of the blood vessels on the skin, which directs blood to the core of the body. This returns the body temperature to the homeostatic level, which is around 37 degrees centigrade. On the right hand side, on the other hand, on the left hand side, in the left hand box, when there's an increased body temperature, we see the opposite happening. Again, this is picked up in the hypothalamus, in the heat loss centre, and the two actions that take place here when body temperature rises are that sweat glands are activated and secrete sweat onto the surface of the skin. Heat is then lost by evaporation, and that the blood vessels of the skin dilate, and this takes heat to the surface of the skin to enable that evaporation. And again, body temperature is eventually restored to the normal level of around 37 degrees centigrade. So thermoregulation acts to redistribute blood between the core and the periphery of the body, depending on the ambient temperature. And this diagram illustrates this rather nicely. When we're warm, the blood is redistributed across the body to the surface of the body, and when we're cold, the blood is retained mainly in the core of the body, and that's to heat the vital organs like the liver and the heart and so forth. And this is all enabled by the capillaries in the skin, which adjust the amount of blood they carry according to the ambient temperature. This is illustrated here, not very clearly, but if you look carefully when we're cold and compare that with when we're hot, look at the relative diameters of the capillaries at the surface of the skin. They're much wider in diameter, more dilated when we're hot than when we're cold. So heat is then lost from the body by evaporation, radiation, and convection and conduction. These are all different mechanisms helping us to lose heat. And in fact, we use some of these mechanisms to gain heat when we're cold. And this diagram illustrates the various ways in which heat can be gained and lost from the body. We can gain it by radiation from the sun or from other warm bodies like radiators, and we can lose it or gain it by conduction, by standing on the ground or in contact with any cold surface. We can lose heat by respiration and by perspiration, and this is through evaporation. This is by the water on the surface of the body or in the lungs being evaporated into the atmosphere, taking the heat away with them. And we can also lose heat by convection. This is by natural breezes, or in the case of being indoors, this is the idea behind using a fan to create a convection current to carry heat away and cool us down. Next, again connected with the skin, is wound healing. The skin is capable of repairing itself when broken, And wound healing takes place in 3 stages. Inflammation, which is linked to the protective mechanisms I'm going to come to later when we look at immunity. Proliferation and maturation. Now again, these are not exactly discrete, they're overlapping phases. So when a tissue is initially injured, you get inflammation. The purpose of inflammation is there to bring cells and other chemicals to the site that will help to initiate wound healing. Then in the middle, tissues remodel, new blood vessels are formed, and finally the wound is completely healed. That's a very simple diagrammatic representation, of course. But here you can see the stages and how long they take. Inflammation lasts about 3 days. Inflammation is a good thing. We tend to think of it as being a bad thing, but in fact it's a good thing, and it leads to heat and swelling and redness at the area. It shouldn't last more than 3 days. If it does, then there may well be infection present. After the inflammatory phase, there's a proliferation phase of about a month, and then finally the wound contracts, collagen accumulates in the wound to remodel it, and it matures, and this can actually take several years. And blood clotting or hemostasis also takes place to prevent blood loss, and this is clearly a major protective mechanism. Before moving on, you might wish to look at this Bozeman Biology YouTube link on blood clotting. Blood clotting is fairly complex, and this is a very simplified version of blood clotting. There are 2 systems, the extrinsic system and the intrinsic system. The extrinsic system works very quickly at the site of injury to produce a blood clot, and the intrinsic system works over a longer time. So the extrinsic system and the intrinsic system are slightly different systems, but they exist on the one hand to give a quick response, and on the second hand, in the case of the intrinsic system, to give a fairly sustained response. They both lead to the formation of prothrombin activator, and prothrombin activator is an enzyme which converts prothrombin, an inactive form, to thrombin, which is an active form, and again that's another enzyme which converts fibrinogen to fibrin, and this combines with platelets to form a blood clot. The process of blood clotting, as I say, is much more complicated than shown there, but it's a cascade mechanism whereby a very small stimulus at the site of injury can cause a very large response. In fact, it's an example of positive feedback. These examples are very rare in physiology, but this is an example, and it stops, or it should stop under normal circumstances, when the clot is formed. Clearly we don't want clotting to go on for too long, or this can also cause problems in the bloodstream. So our clotting mechanisms are under very close control. So here it is diagrammatically. There's an injury, blood leaves, and the injury there and the inflammation initiates the clotting mechanism, and at the bottom we can see ultimately the initial clot, which is activated platelets or combined with fibrin in a network, and that stops the bleeding at the blood vessel. And this is a scanning electron micrograph of a network of fibrin and the red blood cells trapped in it, forming the clot. So just to note, we're going to look at inflammation. It's a defensive mechanism which brings cells to the site of injury, which help to fight the infection and also stimulates wound healing. It's a necessary process. Inflammation can be recognized by redness, heat, pain, and swelling, and also sometimes, if it's extreme, loss of function. In fact, it's often in some injuries mistaken for a fracture of a bone, because all of these things will occur when you fracture a bone as well. Before we move on, you may wish to look at this Bozeman Biology YouTube link on the immune system. In immunity, two processes are involved. There are non-specific defences and specific defences. The non-specific defences are the skin and cells called macrophages. The specific defences are cell-mediated immunity and antigen-mediated immunity. So let's just rehearse the different types of blood cells. There are different types of cells which are described as white cells, neutrophils, eosinophils, basophils, lymphocytes and monocytes. These are all white blood cells. Platelets are concerned with clotting and the red blood cells are concerned with oxygen transport. We'll be concerned here from here on in only with the white blood cells. And just to remind you that these are all formed from a pluripotent stem cell, the hemocytoblast, which produces all these different kinds of cells. The white blood cells, or the leukocytes, are subdivided again into granulocytes and agranulocytes. And the agranulocytes are the lymphocytes and the monocytes, and the granulocytes are the basophils, eosinophils, and the neutrophils. And this just depends on the way that they stain. So first of all, let's look at macrophages. These are literally big eaters. And here we have a macrophage which is engulfing bacteria. The monocytes and the neutrophils are the macrophage cells. Again, what you see here are pseudopods at the bottom being formed by the macrophages. These extend out into the surrounding tissue or fluid, and they, in a sense, capture foreign bodies and engulf them, and then they're broken down inside the cell. So here's the non-specific aspects of protection. If you have injury, you get invasion by foreign bodies. This leads to inflammation. This brings the macrophages to the scene. They ingest the foreign bodies. One of the outcomes of that is the formation of a substance called pus. But also they play a very important part in stimulating the specific immune system, because the macrophages present the antigens to the immune system. They're also called APCs, or antigen presenting cells. So macrophages are also known as antigen-presenting cells. They present antigens to the immune system. This is quite complex, but we'll try and simplify it in the next diagram. And here's a very simplified diagram of how this takes place. The thing to note is that the antigen-presenting cells present the antigens on the surface of the cell. There are two pathways. One is exogenous, whereby extracellular pathogens, for example foreign bodies, bacteria and such like, are ingested into the cell by the action of the pseudopods and the macrophages, but also the endogenous pathway when the cell is infected with a virus. In both cases, the foreign particles are broken down and some fragments are presented on the outside of the cell by the MHCs, or the major histocompatibility proteins. We don't have to worry too much about these, but the basic mechanism is foreign body inside the cell, broken down, fragments are presented along with major histocompatibility proteins on the surface of the cell. The idea here is that on the surface of these cells there has to be a non-self and a self protein presented in order to stimulate the immune system. And here we see a macrophage, and the T cells, which are part of the specific immune system, are reacting with it, and what they're doing is picking up the antigens on the surface of the cell and then initiating their response. And here's that shown diagrammatically, and there are two responses: either mature cytotoxic T cells, which go out and kill other foreign bodies in the cell, or helper cells are produced. And this is the beginning of the specific immune system. So the specific immune system is stimulated when the T cells encounter an antigen-presenting cell. And here's the specific immune system summarised again in a fairly simplified form. Invasion by antigens presented on the outside of the APCs stimulates the lymphocyte system. There are two aspects to this, the T cells and the B cells. These are both produced. T helper cells are produced by the T cells, and the B cells are there to form antibodies, which we'll look at shortly, and the antibodies combine with antigens to form immune complexes, and the T helper cells help in this process, so the action of the B cells requires the action of the T helper cells. The T cells produce killer cells which just ingest the antigens and kill them, and the immune complexes formed with the antigen-antibody complexes are ingested by the eosinophils, which are another of the white blood cells. In both cases, memory cells are formed, and that's the basis of how the immune system can then react to future infections by some antigens which they've already encountered. They don't have to undergo the process of initiating this specific immunity again. In many cases, we already have that immunity, and often we have it for life. So it's also capable of remembering previous infections by the production of antibodies. This is a very complex and exquisite process. And here we have a highly diagrammatic representation of an antibody. They're Y-shaped molecules with heavy chains and light chains, and two light chains at the top. There's a constant region, which is the same for whatever class of antibody we're concerned with, and a variable region at the top which varies, and it's that variable region which is acted upon in specific immunity and is there to help us recognize future attacks by the same antigens. So here we see an antibody at the top, and it's combining with an antigen, either singly or in pairs, forming complexes. In either of these cases, the antigen is neutralized, can have no further effect. These are antigen-antibody complexes that are ingested and destroyed by the eosinophil cells. Now, looking at the specific immune system takes us on nicely to immunization. We can exploit the immune system to our advantage to protect against infectious diseases. And immunization works by presenting an attenuated, this is a slightly damaged or slightly less active form of an antigen, to the immune system. And it's got to be one that also stimulates the production of antibodies. And usually a second presentation of the antigen is required. Now, the specific patterns whereby different agents are administered, in other words, when they're first administered, and the timing between administrations differs for different types of immunization. However, this is the process in a nutshell from the first administration. The primary stimulus would be the first administration, an inoculation. After about 10 days, we get a production of IgM, which is one class of antibody, and also production of another class of antibody, IgG. That's called the primary response. However, look what happens at the secondary stimulus when another dose is administered. You get IgM produced again, but in similar or even smaller quantities than during the primary response, but the IgG antibody is produced in massive amounts, and this is what confers the immunity upon us. The B memory cells have remembered that we've had that antigen presented to us before, and that's what stimulates this large production of IgG. And as I say, sometimes this immunization can be for life. Immunity can be classified in various ways. The large subdivision under immunity is active or passive, and under both active and passive there's natural and artificial immunity. So starting at the left, active natural immunity is when we have a disease and the body produces antibodies to it. Artificial active immunity is when we deliberately introduce a toxoid or a vaccination into the body. This is the process we're familiar with in either being taken ourselves or when we're taking our own children along to the clinic for immunization. Passive natural immunity is when we gain antibodies from our mother in utero, and artificial passive immunity is when we are administered antibodies from another animal or from another human being, as opposed to antigens, and those antibodies help us to fight the disease. This doesn't confer any kind of immunity on us thereafter. It simply deals with the disease at that particular time. So let's recap. We live in a hostile environment and require protection against oxygen, extremes of heat and dehydration. We also require protection against infection. So now some questions. 1. In addition to injury and poisoning, what else do we require protection from? 2. Where are the symptoms of oxygen toxicity manifested? 3. In a reflex arc, what is the correct order for spinal cord, motor neuron, muscle and sensory neuron? 4. Name 4 things that can stimulate the vomiting centre. 5. What 4 things does the skin protect against? 6. How is heat lost from the body? 7. Name the 3 stages of wound healing and in the correct order. 8. Briefly describe blood clotting. 9. What are the cardinal signs of inflammation? 10. Name the components of non-specific and specific defence systems. 11. Describe active and passive immunity. Now you will find some of the answers to those questions in the lecture that I've just given, but I don't expect you to find them all there. I would expect you to look up Google yourself or your own textbooks. Some of the answers can also be found in my blog, 4 Things About, a simple approach to anatomy and physiology, specifically under the entry on protection. Thank you very much for listening.